Hello everyone and welcome to the Medicine Makers video discussion on what has the past taught us about the future, a perspective on COVID-19 vaccines. My name is Stephanie Sutton, editor of the Medicine Maker. I'm delighted to be introducing this event. I'm joined by a fantastic panel of experts, including Adrian Wildfire, who will be moderating the discussion. Give a little background on Adrian. He's Director of Scientific and Business Strategy at hVivo, as well as an accredited specialist in virology, medical microbiology and parasitology. And he's also a subject matter expert in controlled human infection modelling, particularly for diseases of the upper and lower respiratory tract. Adrian also co-founded Chime Agents with Dr. Martin Schuiten, serving as CEO to manufacture wild-type challenge agents for academia and industry. He has authored many papers and articles relating to HIV, ethics, immunology, human challenge models, and many others. I'll now hand over to Adrian to introduce our elite panelists and get the discussion started. Adrian, over to you.
Thank you very much. I'm very excited to be here today. We're going to discuss really what the past has taught us about the future. We're going to look at a perspective on COVID-19 vaccines, and I think that is fairly apposite given where we are, well into the second wave of this particular pandemic. And we have with us today a number of experts. We have two regulators here who've contributed much to the world in terms of giving us the boundaries which we can work in. People like myself who've worked both in general pathology and also in CROs. Pieter's a native of Belgium, trained as an MD and a general practitioner, member of the Worldwide Network on Vaccine Promotion, nominated Associate Professor of Nammo University. He started his own consultancy company, Vaccine Advice. He's the chair of the Human Vaccine Committee of IABS. So thank you very much for coming along, Pieter. We also have Rebecca Cox, Professor of Medical Virology and Head of Influenza Center at University of Bergen. I know she advises the WHO and has been instrumental, I think, in much of the Norwegian Research Council and Department of Health in guiding them in their infectious diseases portfolios. Marco Cavaleri, I've had many lovely initial discussions with, as he is the Head of Biological Health Threats and Vaccines and chair of the EMA COVID task force. So I think he's particularly well placed to comment on what's been happening with vaccines. Trained as a pharmacologist and spent several years in R&D and joined the EMEA scientific administrator and has worked his way up to the head of section for anti-infectives and vaccines. So thank you for coming on, Marco. And last but certainly not least, we have Professor and Dr. Hoft, Professor of Internal Medicine at St. Louis University. I've talked a lot to Dan and visited over times, enjoyed his great hospitality. He was educated and completed his internal medicine, infectious disease and PhD training in microbiology and immunology. So we share a love of microbiology from the University of Iowa. He was named Fellow of the Academy of Science of St. Louis and now heads up a lot of their immunology work, infectious disease work, specializes in tuberculosis. And I believe all of us have knowledge of human challenge modeling as well, which is one of my areas of expertise. And today we're going to look at what's going on in terms of vaccines, what's coming through the pipelines, what it means for us, and also really what have we learned from looking at the past that's enabled us to move so fast to get to vaccines, which we currently have on offer that are offering, I think, unprecedented levels of protection or vaccine efficacy, we'd call it. Just to put this very quickly in perspective, we have a range of transmission, I was going to say standards, not quite the word, deltas going on across the world, from the US, which is 12.5 million cases with over 250,000 deaths and with 179,000 as of yesterday, down to, if we look at Norway, it's only had 33,000 cases, 311 deaths, which is remarkable, and 655 cases as of yesterday. And the UK is falling somewhere in between, I suppose, with 1.5 million cases, 55,000 deaths, and 15,000 cases as of yesterday, probably falling due to lockdown measures currently in place. So looking at that, and looking at the disparity between those countries, and maybe I could go to Dan first, as one of those areas suffering possibly more than others, although obviously Italy, UK and Spain led the charge originally in the first wave. Why do you think the world was so unprepared for a pandemic coronavirus?
Well, I'd say that in some ways we were very prepared in terms of trying to roll out vaccines very rapidly. It's been unprecedented what has happened in this year since this pandemic emerged. But what we haven't done well is standard garden variety public health measures that are critically important when you don't have a vaccine yet. We need to follow the masking, the social distancing, the hand washing, and it's become too political, these issues, which should not have happened. And it's really created a lot of discord in our country and other countries, and that's interfered with our ability to respond to this pandemic. Another issue, I think, is that we, in the beginning, assumed that this infection would be spread only from people that were symptomatic. And that became a very important incorrect assumption, since we now know that lots of people can be asymptomatic and spread, and people that do develop symptoms can be infectious before they develop their symptoms. So that's why it is even more important to be wearing a mask when you have uncontrolled infection spreading like we do in the US. So that we can interrupt. We don't know who's infected if it can be spread from people without symptoms. I'll stop there.
Thank you very much, Dan. So, Becky, as you seem to be the poster boy of this, what's your comment on that? I would just like to say New Zealand had 6 cases of influenza during their season, 6 cases. So some people seem to know something that the rest of us don't. What is that? Sorry, Becky, you're on mute.
No, yeah. We've had 3 cases of influenza so far this season. So yes, we are quite similar. I think that in Norway, we had good and decisive political leadership. We had clear messaging. There was, the public supported the measures to shut us down. So Norway shut down. The first case was in Bergen was on the 28th of February, and we shut down on the 12th of March, before we had any deaths, before there was widespread community spread of the virus. We also implemented testing very early. We had a very good innovation in testing, led actually by people who worked in the oil industry at the NTNU, the Technical University, who found ways of being able to test a lot more people a lot more rapidly through using techniques that they used in the petroleum industry. But ultimately, I think you need good political leadership, you need clear messaging, and you need to have the population behind you. And what we see now is that not only was Norway good initially, we had no cases in Bergen, which is the 2nd largest city, from May till August. And as soon as they started to rise, then they shut us down again. They got it under control. We started to increase again. We're in a sort of mini shutdown again, and now we're down to about 20 cases a day. So if you had clear leadership and you got it under control early, it's much easier to counter this second wave, because you're going from almost 0 and then trying to get it under control. But in many countries, the UK, the US, because it wasn't under control, I think that, there's been a lot of virus spreading generally in the summer. As soon as it gets colder, we come together more, then it becomes very difficult, as Dan has explained, to control. We have not actually had to wear masks here until recently. So social distancing, good hand washing was very important initially, and an early and decisive shutdown, with also good economic support for people who were not able to work. And clearly, if you have good support for people, then they're likely to follow the rules.
And I think we have seen that in the UK and other countries as well, of trying to put these economic measures in place so people have jobs to come back to. Marco, I've got to put you on the spot on this one, talking about February the 28th. In February, I believe the EMA said Europe was fully prepared and it was unlikely that this pandemic would spread across Europe. Now, obviously, that's not from you. Obviously, it was something put out there and was probably fairly well thought through and accurate at the time. Was it a great surprise, and how did the EMA respond to that initially?
I guess, nobody was really prepared for a pandemic like this one with a brand new virus. That's the truth of it. And I think we are all confronted what to do in such a scenario. Even if I have to say that we as EMA, we have been participating with WHO and other groups into looking how we can be prepared for disease X, so the unpredictable, which is what happened here. And unfortunately, you know, this process got a bit stopped in the midst of a real case occurring with this pandemic. But I think we had already revised our health threat plan, so we were ready on how to cope in front of anything that would be emerging, for which maybe it was not related to influenza, because of course for influenza we have really a dedicated plan which is quite detailed and precise into what we are going to do and how we're going to tackle, including the very important aspect of what we can do before the actual pandemic with flu. But here we are talking about a brand new virus, so it's a bit of a different story. And still we were able to activate our plans, which means setting up, you know, this task force and start talking to developers of therapeutic, repurpose or new, and also to start talking about how to advance vaccines as soon as possible. And in the context of vaccine particularly, we already exploited what we were supposed to be discussing more on a theoretical ground, which is how we can bridge data from platform technology from one pathogen to another. And I think that in the lesson learned after this pandemic, there will be a lot of reflection to be made, which are quite important also for the future. So this has been put in place straight away. And of course, I have to admit that one of the lessons learned is indeed, you know, as I was involved also with WHO in the context of the R&D blueprint activity of MERS, particularly for which, unfortunately, we don't have vaccines available as yet, and very few advanced just to phase 1, and also for therapeutic, not a lot of knowledge, and for SARS-CoV-1, pretty much the same. All the development efforts aborted rapidly after the epidemic was out. And one of the learning is that if these efforts were going ahead, probably we would have been much more advanced, at least with vaccine, in terms of having some platform technology for which we would know a lot. And then we could have gone much more rapidly into developing vaccine for this new coronavirus. So that's, in my view, will be one of the other important aspects of the preparedness activity. We need to work in peacetime on what is out there and try to come up with a number of vaccines for a number of these pathogens that may cause outbreaks in the future.
Sure, I think you make a good point. You know, unfortunately, the prodromal period meant there's a long episode of time before, when you're infected, before you become symptomatic, and a lot of people still asymptomatic, anywhere between 40 and 80% between non-mask wearing and mask wearing societies. That's a long time to spread the virus. I think that long period allowed for seeding, especially into areas that have high population fluxes, such as, you know, we've seen in New York and in London, and also we saw in Paris. I think that was a problem. So I think that was one of the issues, and also the fact, you know, it's respiratory pathogen, it can spread very quickly. Peter, do you have any insights as to why we were slightly unprepared, considering the World Bank had pointed out in 2013 and WHO had pointed out in 2016 that beta coronavirus could be problematic?
I think we were not prepared because the previous pandemics, SARS and MERS, were far from our bedside. That was one. The second one, the 2009 H1N1 crisis, has been judged as a mild influenza. So why should we have all these measures in place for such a pandemic? The regulators were expecting an H5N1 crisis, which would have a case fatality rate above 2, above 5, maybe 10, maybe 15. And so everything was linked to a very severe crisis with influenza. But SARS, who was thinking about SARS? That SARS would become a pandemic. And I think that, can you imagine that in Belgium, 6 months before the crisis broke out, the army destroyed something like 15 million masks because we don't need them. Yeah, you can imagine that has been quite deleterious. And I think that is one of the things that we could not imagine, that it would be something like the Spanish flu in 1918. We had a very nice presentation of Arnaud Marchand, and he found a photo of 1918, and it is as if you are back in 1918, 6 people all wearing a very nice mask and social distancing. So the world hasn't changed a lot, but 1918 is a long time ago for most of us, and thus the lessons learned from that were lost. I think the lessons learned from the regulatory affairs, and there I fully agree with Daniel, is that we have a vaccine in the pipeline, or 3 vaccines in the pipeline, before a year after the outbreak of a new crisis with a new virus. This is unprecedented, and I think we learned quite some lessons from the previous time, and I think we are better prepared for getting a vaccine on the market.
Yeah, I think some very good points there, actually. I think we took our eye off the ball, possibly in 2008 when funding for beta coronavirus took a nosedive because we went into an economic crash. So, you know, there wasn't the money and we took our eye off the ball. Economic was the pandemic at the time. It was an economic pandemic. 2009 didn't seem so bad. 1918 was a long time ago. And we were expecting, I think, an influenza outbreak, probably an avian influenza. So I think all very good points. And I think looking back to previous pandemics, you know, as you mentioned, Spanish flu, although it's called French flu, German flu, it depends which country and as to who gets blamed for it. It probably was from the trenches in the UK, but anyway, sorry, in Flanders, etc., but from the UK troops. Hong Kong flu, swine flu, and I suppose I'm just old enough to get the Hong Kong flu in there. How do you think they've influenced modern day vaccines and vaccine manufacturing processes, considering we did get them so quickly in place? We had learned lessons from Ebola. We had learned lessons from HIV. We'd learned lessons from a lot of different outbreaks, epidemics, pandemics, and everyday vaccinology as well, slowly moving forward. So maybe, Marco, you can answer that, because I think the EMA were very percipient in some of their announcements on how we should proceed in developing these vaccines. So what did we learn, and how did that make you, in your considerations, move us forward?
Well, I think for pandemic influenza, indeed, you know, since several years, the EMA has put up really a plan for preparedness in order to have vaccines that can be then rapidly approved once the pandemic strikes. And this is based on the fact that for influenza, we anyway have, first of all, the use of vaccines for seasonal influenza, and the platform are already tested also in terms of efficacy and safety in that regard. But on top of this, of course, in order to tackle a brand new strain for which population can be broadly naive, and for those sparing reasons, the use of adjuvants has come in as an important factor that could help. And this was, in essence, the basis for the majority of the pandemic influenza vaccine that were prepared and used in the pandemic 2009. So what we've seen there, that indeed approving upfront these vaccines based on immunogenicity and safety, and taking into account a strain that has the potential to cause a pandemic, even if of course is not for sure, but still it would be a good proxy for a pandemic strain, was a good strategy, because then we don't have to go through all the various phases of development, particular clinical development, and we could approve just with quality data and some limited data maybe on immunogenicity if at all needed. So that, you know, the vaccines can be deployed really rapidly. But one of the things that we saw in 2009, that even with this plan that was very well prepared in advance, still, you know, the vaccine came just about when the wave was striking in Europe. So we had a reflection around that we need to be faster even than that. And that's why it's so important to look into platform technology that can be much faster than the traditional one, like, you know, based on eggs. That could deliver, you know, large amount of doses of vaccines in a matter of weeks, and not months, as is happening in 2009, 2010. So in the context of pandemic, it's really important to have preparedness plan, advance vaccines for a number of diverse range of pathogens in peacetime, but also thinking about technology that then you can scale up very rapidly once the new virus or the new pathogen is coming up and causing an epidemic or a pandemic, so that you can then deploy the vaccine in ultra rapid time. Otherwise, you will always be too late.
Yeah, I take my hat off to the FDA, WHO, EMA, and many of the other regulatory bodies that said, let's concentrate on safety. Let's make sure what we're issuing, we're producing, doesn't add to any vaccine hesitancy through adverse events, injection site reactions, etc. Let's get something which is safe, and we'll concentrate on escalating the efficacy to move forward. Now, it seems we've had a breakthrough fairly quickly, if we see the news. So I think that's really helped. Dan, do you also think that the EMA, FDA, etc., have taken a supportive approach in this? And in terms of our novel technologies, how do you think they've helped us bring those on to the point where we're actually achieving quite high levels of VE?
Well, I think they certainly have been very important partners in vaccine development, and they are helping all of the rest of the people in the world working on vaccine development to focus on both safety and efficacy. And both of those things are critically important for what you've already mentioned, and that is that there is a lot of vaccine hesitancy, perhaps more here in the US than some other countries. And if we don't do a good job making sure that we do everything we can to make these vaccines as safe as possible and as efficacious as possible, we will lose more trust, and that will become a bigger problem. But I do think in influenza specifically, all of the outbreaks that we've suffered over the years has been something that has driven us to innovation. And that's been very important for vaccine development. It's really ratcheted up in the last 10 to 15 years in terms of, as Marco said, developing platforms that are amenable to rapid rollout for plug and play kind of concepts where you can take a delivery system and put the right antigen in from a new pathogen into it in 2 days in silico, and then within a couple months have a vaccine that's ready to go into people. It's just amazing what has happened. And I think the scares and the frankly weariness from being always concerned about the next influenza pandemic has stimulated a lot of new innovation, thinking about universal influenza vaccines, which we don't have, of course, yet, but there are a lot of concepts in the works that may be able to make some big breakthroughs here in the not too distant future. So I think innovation is key, and influenza recurrent outbreaks and pandemics have clearly pushed the world into this innovative phase, which is very important for our future.
Okay, Pieter, I'd like to go to you, Pieter, actually, because I know you've got a knowledge around mRNA vaccines per se. Were you surprised by some of the vaccine results that previously had been successful in oncology but hadn't shown as much promise in infectious diseases? And do you think using maybe human challenge in the future or using head-to-heads will be a way of testing platforms to see which ones can be advanced? So over to you, Pieter.
I wanted to say one thing in addition to the partners who have worked together. We seem to forget the national competent authorities in Europe, because MHRA, Poleli Institute, the Belgian agency, they all applied these platform technologies in order to start up the clinical trials phase 1, phase 2 much more rapidly. And we talk here about months and not weeks. That is one thing. Another thing is that also these agencies have cut their timelines. If you want to have scientific advice at the Belgian agency, normally you need to submit 7 weeks in advance. Now it is 4 weeks in advance. They really worked very, very hard. And I did quite a lot of these scientific advices, and I must say they were pretty well prepared. Coming back to your question on messenger RNA, Adrian, I think I was a believer, although there were no data available yet on large scale. But why wouldn't it not work if it does work in a lot of animals? In oncology, we see really immunology. But I'm very, very happy that Pfizer, Moderna are coming up with such a nice data, because this opens something for the near future. Because as you said, we will be able to produce quickly, and it will help us if there is another influenza breaks out, then we will be able to use this technology very rapidly, and probably we will be able to do it before the first wave strikes. So personally, I think that was a little bit by hazard. If we go to the vector vaccines, there we were working with a number of companies. EMA has approved over the last months, it was not years. The Ebola vaccines were licensed end of previous year, early this year. And that were vector technologies. And this vector technology has been used by J&J and AstraZeneca to make their new vaccines. And again, platform technology has helped them to skip, but not skip, to skip time, to start earlier with phase 1. And that has been a tremendous gain in time in the development of these vaccines.
Thank you very much, Pieter. Look, in the interest of time, I'd like to hand the next question over to Becky. And that's about what lessons on pandemic preparedness can we take from COVID-19 and previous pandemics? You know, what do you think was ignored, and what can we now put into our preparedness for the future? So how do you think our learning has moved on from this, and how can we be more timely? Because I know you're a great believer in the timeliness of this, and I think you've been proven correct, I think.
I think that there's many important lessons we can learn from COVID, and I think there are important lessons that can also be perhaps applied to seasonal influenza. You know, particularly in Europe, we've had severe problems with nursing homes, and I think that, you know, there are many lessons we could learn in how to protect the elderly moving forward. I think in a way we were a little bit negligent, because we did think, you know, the swine flu was not so severe. We had vaccines, we had therapeutics, and never in my lifetime did I actually think that, you know, we would shut down, and we cover a population of over 1 million people, and we shut the hospital apart from COVID-19 cases. So there are many important lessons, and clearly having plans was important, but the UK and the US were much better prepared than Norway. So having plans is important, but you need good, decisive political leadership. I will come back to that. I think you need politicians that are willing to actually put the population and protecting their population central to their role. And if they don't do that, I believe it is a negligence. So I think that we have learned many things in how we can control, using actually very simple, as we've heard through the discussion, you know, techniques that were used in Spanish flu can also be used now. And we saw after the swine flu that there was a reduction in general infectious diseases, and many of these lessons we can use moving forward. But I think it's a combination. I think, and I would like to say, you know, that I think the synergy we've had through the scientific endeavors from the world and showing how people can globally collaborate has been really important. At the end of the day, it will be the knowledge that we have gained and the amazing achievement of actually running clinical trials, phase 1 and phase 2 together, 2 and 3 together, that will actually ensure that, you know, we will combat this virus. Many important lessons, and many important lessons for leadership to, you know, good leadership I think is essential. So you can have the best plans in place, but without good leadership, we see the consequences can be dire.
And I think the synchrony and cooperation has been remarkable to everybody, you know, through everything in terms of both economic, through to PPE, due to manufacture of additional respirators, clinical care, trial of drugs, emergency authorizations. I think that coordination and cooperation has been absolutely fantastic. I think lessons we've learned in terms of drugs from HIV, helped us in hepatitis C. I think lessons we've learned from influenza have helped us in the beta coronaviridae. I think, this is a great time really for science to actually try and shine and show us the way out of that. And I think, some of this has been exemplified by the speed of the vaccines that have come through. So, Dan,
can I just say one thing quickly? I would like to say, the work done by the WHO, I think, has been really important. For example, in their solidarity drug trials, where you see when you actually get the larger number of patients, then you can actually see whether antivirals are effective, and also in their hope to move forward with vaccine trials. So I think there's been a lot of very good work done by the global health agencies. Sorry, Adrian.
Indeed. No, I think that's a very well made point, actually. And I feel in some ways we haven't just relearned the lesson, but we've reaffirmed those lessons. We now understand more about mucosal immunology and how important that is. We see IgM waning after 3 months, which is when you start to see some more increase in susceptibilities. We've seen these big drug trials show that actually getting rid of virus when people are severely ill, they're hospitalized, they're on respirators, doesn't make a great deal of difference, whether it be hyperimmune serum or monoclonal. And those are more prophylactic treatments. I think we know more about when we should treat with antivirals and when we should treat with, say, anti-inflammatories. I think we've learned that very well. And I think it positions us better in the future going into other diseases. It's taught us that timing can be everything. Dan, what are your take-home messages from what we've seen in the past? Let's talk about vaccines, especially.
I'll get to vaccines, but I'd like to support Becky in saying that I believe thoroughly that it's essential that leadership is decisive, focuses on this, that there's a single voice, that it's a rapid response. And another issue is that you can't think of public health separate from the economy. Those two go hand in hand, and you cannot improve the economy in the setting of a pandemic unless you really focus on the pandemic. So that is a critically important lesson, I think, that we've learned. In terms of the immunology of vaccine development, I believe was your next question. I do agree with you that we need a lot more research in areas of mucosal immunity. That's a kind of a frontier for us still. We have some vaccines that can induce presumably good immunity through delivery, such as the flu mist and other vaccines, but we don't know enough to really know how to harness its potential. I think the other area in immunology that we don't know enough about is the role of T cells. We know that they're critical for helping the B cells develop the best neutralizing antibody responses and other types of protective antibody responses. We know they're critical, that the CD4 positive T helper cells are critical for helping CD8 killers to kill virus infected cells, but we don't know enough about how to coordinate all three components, the CD4 cell, the CD8 cell, and the B cell together to get the best combination of responses. And that's an area that is very important for future investment.
Okay, before I give it to Marco, I'd just like to add that I think we can learn from the H1N1 pandemic that actually without large herd immunity and immunological pressures, whether that comes from direct infection, natural infection in the community, or immunization, there's not much pressure on some of these viruses to change antigenically. And we didn't see H1N1 change very much. H3N2 is a different matter, but H1N1 hasn't changed a huge amount. And I think with the beta coronaviridae, we've seen D614G change, we've seen other minority changes which don't seem to make a great deal of difference to the immunogenicity. So I have a feeling that these vaccines, much like the H1N1, which has shown 60% efficacy quite consistently, that we're probably going to see quite consistent efficacy with this vaccine we're producing now until there's enough herd immunity to make vaccine escape of some mutations possible. So, Marco, you've got a good smile on there as if I'm talking rubbish. Tell me why I'm talking rubbish.
No. I like your optimism, and probably you're right. But of course, we as regulators, we have to really monitor careful what is happening. Like, you know, these new variant 5 that emerged in Denmark, you know, we need to understand what is the impact on the neutralization from the different vaccine. And it does not seem to be a problem, but of course we have to be careful and monitor and surveil, have a good surveillance to understand what kind of variant may be emerging in the future, and whether indeed we may lose the protection from some of these vaccines. Now, these vaccines, they seem to be quite potent, and so they might also get quite a good cross protection or cross neutralization across different variants. And but we need to characterize that very well. And that's why we're asking manufacturers to test sera from vaccinated people against any new emerging variants, so that we got a good overview and a good overlook at what is happening there and whether there is any chance that we might lose some of the protection. But I also completely agree with what you and Dan and Becky said about the role of T-cell responses as well. Clearly they have a role. Of course, here we're talking about different endpoints in a way, because we want to prevent mortality and morbidity. So really the infection deep in the lung and the replication deep in the lung, and then the sequelae that from this derive. But also we want to prevent, you know, really viral replication in the upper respiratory tract, which is linked to transmission, and then you know, is very important from the public health perspective. And for that, you may need, you know, different kind of immune responses coming from mucosal antibodies, neutralizing antibodies, and the role of T cells might be differently important according to what you're trying to protect from. We need a better understanding, but we know that when it comes to T-cell responses, it's always been difficult to have something that could be validated or be used as a correlate of protection. Unfortunately, we are still stuck with that. And of course, we are not stopping to look into what we can do with this, but you know, history has been very clear that so far only antibodies have been a possible way of determining something that could be used as a correlate of protection, or something that as an immune marker could be used for immunobridging, which of course is one of the big topics now when thinking about next generation COVID-19 vaccines.
Very interesting points. And I think, Dan, maybe you would have something to say. We're going to have a really nice sort of final topic in a minute where we're all going to have a bit of fun. But Dan, just to finish up very quickly on that, I mean, you're a T-cell man, you're a tuberculosis man, Th1 runs in your blood. How do you think we can measure these T-cell responses going forward? Because our previous techniques like quantiferon gold and things like that pretty much hammers to crack a nut. Do you think we're going to get better at monitoring T-cell priming going forward from these, both these new vaccines and from the new learning of the immunology of COVID?
Yes, I think we are already getting better, and that you already mentioned the quantiferon test, the IGRA test, the interferon gamma release assay tests, and for TB diagnostics. Those are well standardized for a Th1-like response, looking at interferon gamma responses by the CD4 cell predominantly, but the newer generations also look at some CD8 response. There are new tools that I think are very promising. The work looking at TCR signatures is potentially very amenable to looking for diagnostics. There's already some early work and preprints that I've seen that are suggesting that those might be very useful for diagnostics. There's much more that we're learning about how to trigger the best CD4 Th1 response and how to get a better CD8 response with vaccines. So there's rich areas for continuing research that I think will pay off in the near future to help with these challenges that we have.
Thank you. Thank you, Dan. Okay. We've got sort of 5 minutes left, and I think we're going to get a minute each if we're lucky on this last one. And I'll start with Pieter. So when it comes to COVID-19, are there any current projects you're particularly excited about? And you may mention some of the new hopefuls. Sorry, you're on mute, Pieter.
Sorry for that. I already mentioned it. I'm so excited about the messenger RNA vaccines. If they really succeed in doing the job, this is really marvelous, because Stanley Plotkin will have to rewrite his textbook, because we get a lot of new vaccines based on this technology. So that is for me the biggest excitement, that both the first two messenger RNA went over the 90% efficacy. Well, efficacy, let's say good data, very promising data, and let's hope that it continues like that.
Thank you very much. And I think I owe you a bottle of wine, actually, because I was slightly more skeptical. Maybe the lipid nanoparticles made all the difference, and I was way off. So Pieter, yeah, next time we meet, I'm afraid there's a Chateau Margaux or something from me. Let's go to Marco. What are you excited about?
Well,
I mean, don't be too biased, obviously, because you're the EMA, so you'll have to be a bit careful, I guess.
Indeed. My excitement made not as much as Pieter, but of course, you know, it's quite incredible to see how fast these vaccines have been progressing over less than a year. And again, what I've never stopped to say is when people ask me or ask the EMA is that here there were no corners really capped. You know, a huge amount of data being generated, both preclinically and clinically with this vaccine. So it's quite incredible when, you know, everybody's joining forces and the private sector is working with academic sector and with the governments, what you can achieve in terms of at least reaching the stage that some vaccines have already top-line results. So that for me is what is most remarkable, and, you know, I'm really pleased to see. Then, of course, the top-line data that are coming looks marvelous, but of course, we need to assess the data as always. So until this is done, you know, I will not say much more than this. But it seems like, you know, these new technologies are probably here to stay, and really, it's a revolution in vaccinology.
So, very, very quickly, do you think we're seeing a new paradigm or a new development pathway for vaccines? It feels like it.
It feels like this, and you know, and what is really paradoxical, that a lot of people are saying here things have gone too fast, but in actual fact, you know, the requirements for the clinical trials, at least for the pivotal trials, are more stringent than what we normally do. And that's something that I think everybody should recognize. And of course, we will have to think whether this is going to stay, or if we will return to normality and being a bit more relaxed in terms of the acceptance criteria for the pivotal phase 3 trials. Because in some area, it could be a bit of a problem in terms of the number of cases that can be recruited and the efforts that this will mean. But definitely what has happened here is really fantastic, and really shows that, you know, when there is the willingness to do things, when there are the funds available and everybody's working together, really committed, you know, even things that might sound impossible will happen.
Fantastic. Dan, over to you.
So I would agree with Pieter that that the mRNA vaccines are one of the most important things that are coming out of this, and that's very exciting. I would say that I'm looking towards the future. This is going to happen again. It's happened 3 times in the last 20 years. So we have to be ready for the next one too. So applying the lessons we've learned here to try to prevent the next one. And in terms of looking at things like broadly neutralizing antibody targets for vaccines that might be able to work against new coronaviruses, for example. Developing Tcell based vaccines that could work across the pathologic, at least, coronavirus strains. And then I think another thing we've really learned is how important it is, as Marco was mentioning, that there's a partnership between government, not-for-profit, academia, and the business world. When you can combine the ability to bring $10 billion to the table, amazing things can happen in these partnerships. Of course, we won't always be able to do that, but we're seeing the potential. And if we do it in advance, maybe it'll be cheaper in the long run.
Yeah, well said. You know, it's just quite remarkable. We've got an upper respiratory tract virus. We may have a virus in a year. Now, if anyone said that to me, well, people did say that to me a year ago, and I said, you must be joking. 18 months, 2 years. And you know, if I'm wrong, I'm the happiest man to be wrong. So, Becky, I'll leave the last word to you. What are you excited about? What do you think's really come along and kicked us as scientists and regulators and people who are immersed, myself, very much sort of in the business side, and yourself purely in the scientific side? You know, what's made the difference? What makes you excited about this?
Well, like the others, I'm super excited about the vaccines. I'm a little nervous about the rollout, both in terms of will it be equitable and will it be successful? But most of all, if I have the last word, I'm looking forward to the end of this. I think, you know, this has been a once in a lifetime learning experience, and let's hope we learn from it. We continue with the good global cooperation and roll on the time when we're not living in a pandemic.
Thank you, Becky. Thank you very much for that very positive last comment. Look, I want to thank all of you, each and every one of you coming along today. It's been an amazing talk. I think we've really had a quick romp through what we've learned from the history. Maybe we didn't mention Jenner. Maybe we didn't mention how that feeds into maybe Codagenics with their codon deoptimized vaccine. But we've covered a lot of ground. And I want to thank each and every one of you for helping look not just only at the scientific side of it, but also lessons learned that have helped the regulators help us in our work. So I'll wrap up just saying thank you to all, and thank you for everyone for watching.